Hi everyone, my name's Rob Beagrie. I'm from the University of Oxford and from the Doug Higgs Lab, and I'm going to talk to you about my poster Dynamics of the 4D genome during in vivo mouse erythroid lineage, specification and differentiation. So in the Higgs Lab, we're really interested in enhancers because they're a key component of human genetic disease. So enhancers are regions of regulatory DNA that are really responsible for switching on nearby genes, usually in specific cell types or developmental time points. So for example, on the right we see two Belgian families that have inherited polydactyly, and this the allele in both cases is actually not a change in the protein coding gene, but is a single base pair change to an enhancer, which actually in this case is 1 million base pairs away on the linear genome. So even though it's very far in the DNA sequence, what happens here is that there's a DNA loop which is formed in the 3D space of the nucleus that brings this enhancer close to the target gene that it regulates. And one of the key questions in the field really is, how does this looping between enhancers and promoters work? So when are the loops formed? And there are really two models. The first is the permissive model on the left, where the enhancer promoter loops are formed early in development before the gene is activated, and then as the cell differentiates, those enhancers recruit transcription factors or co-activators that then switch on the gene which they're already in contact with. Alternatively, in the instructive model, the promoter and enhancers don't contact each other early in development when the gene is off. Rather, the enhancer looping to contact the promoter is part of the molecular switch that actually turns the gene on in the first place and is a key component of transcriptional activation. So we wanted to look at this in vivo primary cells, and we used alpha globin, which is a really important erythroid gene, in mouse fetal liver cells. So we divide the population of fetal liver cells into several different populations. S0 low is the earliest, then S0 medium, then S1, S2, S3, and S4, S5. So this is the differentiation scheme that we're going to be using throughout this presentation, and these are all primary cells. And the first thing we needed to know was when during this differentiation system the gene is actually first expressed. So we looked at this a number of different ways. This is single-cell RNA-seq, showing that there's a massive increase in transcription of the alpha-globin gene, really around the S2 and S3 stages. But we also see a more subtle increase that occurs earlier between S0 medium and S1. And because this is a relatively small change, we wanted to confirm that this was real using an orthogonal technique. So we carried out nascent RNA fish, where we really fluorescently labeled the intron of the alpha globin gene to count how many cells are actually expressing it at any given time. And what we find is that we are able to reproduce this very early upregulation of alpha globin that starts at around the S0 medium or S1 stage. So having determined when expression starts, we then needed to look at DNA loops. So Marika and Jim Hughes, who are key collaborators, developed this really cool new method called tiled C, which is essentially a variant of 3C, where you fix the chromatin using formaldehyde, you digest using a restriction enzyme, religate the DNA together, and then you capture these 3D DNA contacts. And the way that tiled C varies from other 3C methods is that all of the library prep happens in a single tube, so you maintain complexity of the library and you can look at very small numbers of cells. And we also use biotinylated capture oligonucleotides that specifically pull down every single restriction fragment in a 3 megabase window around the alpha globin genes. So using this method, we were able to really examine in detail the chromatin folding at alpha globin, and in the heat map on the right hand side, the blue colours indicate two pieces of DNA on the linear genome which are not interacting very strongly, and the red colour indicates pieces of DNA that interact quite strongly. So hopefully what you can appreciate is that the interaction between the enhancers, which are annotated underneath, and the two adult alpha-globin genes, which are annotated in red, do indeed start to form at around S0 medium or S1, which is really exactly the stage where we also see transcriptional upregulation. So it looks to us as if the formation of the chromatin loop and the activation of the gene are actually occurring at the same time, so more in line with the instructive rather than the permissive model of looping. And since this is only one erythroid gene, we wanted to look at some other genes which are upregulated in differentiation. And here we quantify the contacts between a gene and its enhancers alongside the expression level of that gene for 6 different erythroid genes. And in each case we see that the dynamics of formation of the enhancer promoter loops and the activation of the gene itself are actually quite comparable. This is for all 6 genes. We see expression occurring at a similar time to the formation of the DNA loops. So all of that was published just a few weeks ago in Nature Communications, so please feel free to go and read that if you're interested. And at the moment we're now using chromatin accessibility changes to try and figure out which transcription factors might be involved in this switch or forming the loops, activating the gene. So we've looked at peaks which change in accessibility between S0 and S1, and we found strong enrichment for GATA1 and KLF1 motifs, which we're now looking to follow up. And with that, I'd just like to thank all of these people that were involved in this work. And please feel free to send me any questions on Slack or Twitter. Thanks.